confirmation that we're started. There we go. The thing just went ding. So we are recording right now, and welcome to our first lab, my first attempt to teach a hands-on lab by remote. And almost everybody in this class has received a microscope except 3 people who are having theirs shipped, and I'm told that they went out yesterday. So we'll start with this, and I want to talk you through the parts of a compound microscope. And again, this is stuff you've probably seen before, but I just want to make sure that we're all starting on the same page. So here's the scope that I've got. And of course, down at the base, you've got the illuminator, and there's a little, down here, there's a dial control that you can use to make the light bright. Duh, okay, this is pretty obvious. The light comes out the lamp and goes up through this. There's a little gizmo right here called a condenser, and the condenser that raise it or lower it. So there I'm raising it, and there I'm lowering it. And on this model, there's a dial that you can turn back and forth. I think some of yours probably just have a lever, but either way, it's a control that you can dial back and forth to open or close the condenser aperture. Back in the days before cell phones, we had these things called cameras, and they had something called an f-stop in them, which was a diaphragm, just some pieces of dark colored metal that you could, open up like this or close down like that. And you can do the same thing here. That's your second way of controlling brightness. Common error that I see is people trying to observe with the iris diaphragm all the way open. And this will make your image very bright, but it also tends to wash out fine detail. So if you can't really see anything because everything looks very uniformly bright, turn that diaphragm down until you can see clearly. The condenser, there's a somewhat involved way to adjust the condenser height to get what we call critical illumination. I'm not going to bother with that here. For your purposes, it's probably best if the condenser is all the way up and then about a quarter turn back down. So you can adjust your condenser by turning that dial to move it all the way up and then racking it back down about a quarter turn, and that will be pretty close to the optimum. Okay, right up here you've got your nose piece. And of course, you've got 3, or in my case, I think I've got 4 objective lenses, and each one will successively click into the light path. You always want to start on low power. And I've seen even students with some microscope experience make the mistake of starting at high power. And the reason simply is that if you're looking for something, and you zoom in with very high magnification, right? It's kind of like trying to find a particular tree in the forest by looking through a magnifying glass, right? If you want to find something like that, you're much better served using something with a much wider field of view, like your own eyes, and then zooming in once you've got that particular tree. It's the same thing here. So start at low power. And scopes are focal, meaning that if you get it focused at low power, then moving to the next highest power, you should be close to being in focus, and you should only have to adjust the focus knob a little bit. By the way, down here is the focusing knobs, and the big one makes large-scale movements of the microscope stage, and the little one makes much smaller scale movements. When you're up on medium to high power, you only want to use that fine focus because if you use the coarse focus, you could slam your slide into the objective lens. And if you break that, that's kind of expensive to replace. And if we have to spend money on replacing microscopes and lenses, we can't spend money on more important things like purple and gray astroturf on the field. You know, priorities here. Okay. The stage also has these two little knobs right here that let you slide it forward and backwards, left and right. I assume you've seen those before. And then, of course, up here at the top, you've got eyepieces. The head of the microscope, there should be a little knob here so you can rotate this to any position that you're comfortable. If you loosen the knob completely, you can take the head completely off, but don't do that because that will get dust into the light path. And also, don't ever pick it up like this, because the last idiot to use the microscope might have loosened that knob. And if you pick it up and try to support the weight of the whole thing by the head, you can end up holding the head with the rest of the body of the microscope falling and crashing on the floor. And we do not want that. If you break these scopes, I'm going to have to look at you very sternly. Something like that. Okay. Common mistakes that I see, again, are just cranking open the condenser too much and you get a bright image but a washed out one. The condenser too high up can also decrease your contrast. If you drop the condenser down, the picture will tend to get dimmer, but it may also be a lot more sharper, a lot more contrasty, and that can be important when we're looking at translucent things, as we will do. Other common mistakes is getting dirt on the slides or dirt on the lenses. Make sure the bottom of your slides are dry. If they're wet, they'll stick to the stage by capillary action, and it's hard to move them. If there's dust on your eyepiece or objective lenses, use that lens tissue that you should have been provided. If it won't go away with that, you can try using a little bit of a solvent. The best thing is like 70% rubbing alcohol. If you don't have that, I'm sure none of you have vodka in your houses, because if you're under 21, that's illegal, and I know none of you would break the law. Distilled water or something like that would also be a good thing to try for starters. You don't want to use acetone because that can dissolve plastic. If non-optical parts are dusty, like this illuminator down here, that can just be cleaned off with a Kleenex or toilet paper. I'm sure your neighbor's got 50 damn rolls in his garage by now, so we won't miss one. So you could clean this off with a Kleenex, a toilet paper, or even a piece of soft cloth. The same thing is true for microscope slides. Those usually don't have to be treated with a lens tissue. You can clean those off with a Kleenex or TP or something very soft like that. But yeah, they're not quite as delicate as that coated optical glass that we have. You've been given a box of slides, and you'll notice they all have the word up or this side up on them. Make sure that the side that says up is facing up when you open these, because if you don't, these slides all go falling out and some of them break and the rest have to be put back, and sorting them is a colossal pain in the neck. I, of course, have never done this, but heavens no. But some of you young, junior woodchucks might make that mistake. So please, in the name of all gods that might be listening, make sure that the upside is in fact facing up when you open the slide box. There's always somebody. Don't let it be you. The slide box has slides that, of course, look like, look like this. You've seen these before. And then again, those can be cleaned off with ordinary soft towels or toilet tissue or Kleenex or something like that if they get a little bit of dirt on them. What now? So you'll note that this scope, mine happens to have two eyepieces. Most of you got scopes with just one eyepiece because Dr. Hill was being cheap. A few of you got scopes with two, and if they've got scopes with two, you can swing the eyepieces closer or farther apart to match your own pupillary disc. They tend to be really wide. You can do, set it to fit your taste. One of my eyepieces has been replaced with a little camera, which will enable me, just a moment to set this up carefully, properly, and away from the cat. Once I get this, why, cat? Why? What is your... You have been given food, water, and cuddles. What more is that which you desire? Okay. Everybody, can you see something looking different now? Yeah, Shivam, you know what it's like to open these things up with the wrong side. Somebody text me. You're not seeing me now, I take it. You're seeing the field of view in the microscope? Yes? Please, somebody give me confirmation that this is working right. Okay, Autumn says yes. So you'll see something like this when you put a slide under the scope. And I'm going to move it around, of course, and I'm not properly focused. And what I'm looking at is the prepared slide. You should have a very similar one in your own slide box. And this contains skeletons of radiolaria. In yesterday's lecture, we ended by talking about this big group of protists called Rhizaria, and that includes radiolarians, and it also includes forams. It includes some other groups in this grouping called the Cercozoa that we're not going to have much time for. But radiolarians are distinctive because they make these silica shells inside the cell. And they often have this very lacy appearance, and typically they have spines. So you can see the big one right there in the center is one of these radiolarians that is spherically symmetrical. And then over here, you can see one of the ones that looks like a helmet. Be kind of a funny looking helmet, but you can see it's kind of cone shaped and broader at one end than the other. There, so we've got, yeah, there's another fairly large there. These being are broken a little bit, but you still get the idea of what these radiolarian shells look like. There, that one's kind of cool. Just to show you what the optics of this do, if I swing the condenser wide open, you can see that the image gets brighter, but it also gets washed out. You're not getting a lot of contrast there. You're not seeing quite what your eye would see because this camera automatically compensates for brightness differences. So if I turn the light down, you see it flickers a little bit, but the overall brightness stays pretty steady, and that's the camera's doing. That's not what your eyes would see. But you can certainly see that I've got the illumination all the way up, and this looks really washed out with the diaphragm open all the way. So if I close it down a bit, there, it's still bright, but I've got much better contrast. And if I rack the condenser down, well, if I rack it up, Yeah, again, I don't think it makes that much difference, but you'll probably get best results with your eyes if you have it almost all the way up, but not quite. So if I change the magnification, all I have to do is turn that fine focusing knob a couple of times, and there we go. There's a nice little radiolarian right there. It's thick enough that I can't get the entire thing in focus all at once. So right there, I'm focused kind of on the part that's closest to me. And then there, that's not in focus, but we've got, crisper views of other parts as well. So for a lot of critters that we see, you are going to have to focus up and down. So there's another one with spherical symmetry. You can see it's basically a sphere with radiating spines. This is the kind of thing that would just look gorgeous on a Christmas tree. There's a modern jazz group called Medeski, Martin, and Wood that actually released some albums with music inspired by radiolarians. I don't happen to have them, but one of these days I need to check them out because, they're really good musicians, and also how cool is it that they're naming things after radiolarians. So there's another one of the helmet-shaped ones, and so on. And of course, you can and should do this at home with your own scope. You do it right this minute, but you see something similar to what I see. And I'm not going to require you to do anything with it, but I do want to reinforce what we talked about in lecture by showing you the same things with your own eyes. And then if we go to, there's a prepared slide with forams. This will also, just looking at these will give you some practice in, just messing around with the scope and figuring out what you're focusing on can be a little tricky. But let me see if I can find the forams here. Whoops, was that one? That actually looks more like a, okay, there we go. All right, there's a foram. That particular one is probably a planktonic foram. It's one of the ones that float, although they're completely different species that live on the bottom in and among sand grains. In fact, in some places they are the sand grains. But that's a little planktonic one, and you can see it's made up of multiple chambers. In this species, they look kind of like four bubbles stuck together. And the chambers form as the protist grows, and the arrangement can be very distinctive. So if I zoom in on this one a little bit, and noodle with the focus. And now can't find it. I swear it was around here a minute ago. Okay, that's a bit of a broken one. Where was that? How did I lose that thing? Oh, okay. What the heck? That's not the foram I had in mind, but that's a different species. It's got what's called biseriate arrangement of the chambers, meaning that the chambers are, in two rows that kind of alternate with each other across the midline. So you have that kind of zigzaggy shape. That broken one, I don't really know what that is, but this one is just one that's has a different arrangement of chambers. And zoom in on that one. There we go. And now that we're zoomed in, you can see as you focus up and down, those little holes that are present in the wall of the foram test or shell, those are where, when this thing was alive, the little reticulopods came out and joined and split and joined again to make that network that the foram uses for feeding. I've added some footage of a live foram, by the way, to your playlist for this course. You don't absolutely have to watch it, in the sense I'm not going to test you on it, but it does show better than I could do with just static slides what the live critters look like and how they make a living. Okay, are we happy? All right, somebody chat something just to show that, you know, message is received here. And I'm going to put one more. Okay, Lauren says she is receiving. Hazer says looks good. Garrett says good. So, all right. So far, so not entirely horrible. Somewhere, is the cat sitting on it? Oh, cat, what have you done? Why are you being so pestiferous right now? You cat, you. Stentor. Where did I put? Okay. Hey, get off that. Oh, there it is. I just misplaced the slide. All right. Okay. Let's put this in and snoop around a little bit. This is a slide of there we go. Morning, Savannah. Okay, what we're looking at here is a prepared, and this is not nearly as cool as seeing them live, but what these are is a ciliated protist called paramecium, a very common one in biology labs. You've probably seen these guys before because, as I said, I think Steve Carafit usually orders these for class. And if we zoom in, sorry about that, the cat just walked on the okay, the fact that I've got that pattern tells me that what I badly need to do is clean lenses. Let me see if I can fiddle with the lighting and not make it quite so bad. There. Now, where was the paramecium? Okay, so we've got that one in the center. It's not perfectly in focus, but if we tee it up with higher magnification, there. There we go. Live paramecium is pretty big. The biggest ones can get up to about 150 microns. And the overall shape is often compared to a slipper. These have been distorted a tiny bit through the staining process, but they're often called the slipper animalcule, or the slipper organism. And the body here is stained kind of this light tan color. And there, if I close the condenser down, you can see it just a little bit better. That light tan kind of oblong to slipper shaped thing in the middle is a paramecium. And the darkly stained object in the middle of it is the macronucleus. Now, paramecium belongs to a group that I haven't lectured about yet. We'll do that tomorrow. It's called the Ciliophora, or just the ciliate protists. And the exterior is covered in cilia, right? Little short, beating hair-like organelles that enable the ciliate to swim. With a rather complicated little arrangement of cilia. They're put together in a fairly complicated way with a bunch of little fibers anchoring them in a very strict pattern. Again, ciliates are probably the single most complicated single cells that there are. The main thing that you're seeing is that dark red thing in the middle. That's called the macronucleus. Ciliates actually have a unique feature called nuclear dimorphism. That big blob, the macronucleus, is made up of several thousand very short chromosomes, each one of which might only have a couple of genes on it, and each one of which is replicated thousands of times. So this is like a 1,000 ploid nucleus of maybe 10,000 different chromosomes. It's almost like just a giant bag of genes kind of floating around. The details vary a bit from one taxon to the other, but to make a nucleus like that, some ciliates do this really wild feat of DNA cutting and pasting and splicing that's actually got cell biologists very excited of late. There's a lot of new research coming out on the really freaky things that ciliates do with their DNA. Now, next to the macronucleus, there should be a micronucleus, which is much smaller and is much better behaved. The micronucleus is basically a diploid nucleus that follows all of your basic eukaryote rules. But it's very hard to see in these slides because A, it's tiny, and B, it's usually butted right up against the macronucleus. So the macronucleus is easy. The micro is a lot harder to see. I'll try to zoom in on high power here. And I'll try to make that annoying pattern go away, but clearly what I need to do is dust off this scope. Because let me see if I can get this silly thing to focus. Okay, I can't see an awful lot of detail, and the main culprit is the fact that I really need to give this thing a good cleaning, and you probably should with your own scopes. There, I can sort of make that go away, but now I'm kind of losing detail a little bit. So yeah, clean your scopes, and I can't really see the micronucleus in here, but that's not unusual because again, it's tiny and it butts up right onto the macronucleus. You kind of have to be lucky to be able to spot it. So that's what a fairly typical ciliate looks like. I've got one more. This is a different species, and these guys are huge. They can be up to 1 millimeter in size. There were actually some very cool on cell pattern formation by taking these ciliates and actually cutting them apart, doing surgery on them. They're among the few single-celled organisms that are big enough that you could actually do that. And there's one, but it's not shaped. There we go. Okay, this is a trumpet-shaped ciliate. It's called Stentor, and it's the sworn enemy of He-Man. No, wait, sorry, that was somebody different. Stentor was actually a hero of Greek mythology. Yeah, the original Stentor was a hero of Greek mythology who had a louder voice than anybody. He was the Greek herald at in the Trojan War. We still can use the word stentorian to mean something very loud. A stentorian voice is a very loud one. Anyway, I digress. You can see it's got a narrow end and a wider end, and this thing that looks like a red string of beads, that's the macronucleus. The shape of the macronucleus depends on the species of ciliate. There are some that have just kind of roundish ones, some that are horseshoe-shaped or strip-shaped, or even kind of S-shaped or number 3-shaped. And then this one and its relatives have a macronucleus that looks like a big string of beads. So different types of nuclei have different shapes. And if I can get this one focused, there we go. There's a close-up of that bead-shaped macronucleus. And I promise you there is a micronucleus in there somewhere. I didn't make it up. The broad end, it's hard to see in this particular one. I might look for one that shows it better. But ciliates actually have a sort of cell mouth where food particles are taken in. It's called a cytostome. Cytocell, stoma, mouth, duh. And for some ciliates, that's all there is, just kind of little, slit or pore on the outside of the cell where food particles get taken in. But others have structures that surround the mouth or the cytostome, called the peristome. And paramecium has a groove called the oral groove that leads to the cytostome. And there's actually some little specialized cilia in the groove that beat to draw food particles in. Stentor's got kind of a spiraling peristome, but you can't really see it from this angle. So let me see if I can find one with a better angle to show you that spiraling peristome that Stentor has got. Yeah, there's a few kind of little shredded ones right here, but all right, hang on. This is going to go out of focus for just a little bit because I'm going to try to find one using my eyes, and the focal point of my eyes is not the same as the focal point of the camera. So when I'm looking through this, it looks perfectly focused to me, but it doesn't look good to y'all. So momentary bits where all you can see is this annoying blob. Okay, that one looks kind of disrupted. Yeah, that's the problem with the stain slides, is that it's probably impossible to get ciliates that show you perfectly what the structure is and aren't distorted in some way. That's just, the chemicals that you use to preserve these guys also tend to make them shrink up. All right, let me see if I can. There, okay. All right, there's that nameless blur, and the tail end is back there. And the problem is the head end is kind of shrunken in, but kind of there, just back of that tip-shaped thing, you can see this kind of shadowy round object, partly covered up by the macronucleus. I think that's the peristome, which has this kind of circle, or actually better yet, a spiraling inward shape to it. You might see this a little better in your own prepared slides. Anyway, I don't want to get too deeply into that, but almost all ciliates do have a cytostome. Many of them do have a peristome of a particular shape, and identifying those, being able to find them in species, which we'll talk about in the second part of this lab. So I will go ahead and stop recording now.